Good afternoon, ladies and gentlemen. First, I'd like to thank Rajiv and his team very much for the invitation to give you a talk on genomics-based valorization of genetic resources for improving disease resistance in cereals. More than 50% of the worldwide acreage are covered by cereals. And besides rice and maize, wheat with an acreage of 200 million hectares and barley with an acreage of 50 million hectares are of prime importance. Both wheat and barley are mainly used for human nutrition and animal feed and are, as already mentioned, of prime importance for feeding the Earth's growing population. But both wheat and barley are hit by many fungal pathogens, by viruses, and by insects. And on the worldwide level, losses, of course, regionally different, are estimated to around 15 to 20% losses due to diseases and insects. And for wheat, this amounts for around 140 million tons, equivalent to $35 billion. So, breeding for resistance to biotic stress in cereals is of prime importance to avoid yield losses, to ensure a consumer and environmental friendly production, and to feed the Earth's growing population. So if we have a look on the success of breeding for resistance in Germany, we can see that over the last 6 decades, the susceptibility to powdery mildew in wheat was considerably reduced. The same holds true for leaf rust, to a lesser extent to septoria, but nearly no breeding progress was achieved with respect to tan spot, because tan spot is inherited in a more complicated way, while for these diseases monogenic resistances are available. But the problem is not solved, because breeding for resistance is to some extent a never-ending story. A new cultivar is released carrying a new monogenic resistance gene. The acreage increases. This imposes selection pressure on the pathogen. First, virulent isolates appear. They increase in frequency, and the resistance will break down. We all know about this boom and bust cycle. So, it's an eternal battle, because the economic impact of common diseases on cereal production and the ability of pathogens to generate new virulent patotype in a short period of time necessitates searching for new resistance genes, and in parallel, the development of tools for the efficient deployment in breeding programs. So, each harnessing of genetic diversity, which is present in gene banks, which we learned this morning, starts with the development of effective screening methods for resistance, followed by the identification of genetic variation for resistance within the gene. Once interesting genotypes are identified, genetic analysis of resistance has to be conducted to get information: is resistance inherited in a quantitative or in a qualitative manner? And then in the next step, molecular markers have to be developed, and these markers are suited to introgress respective resistances into adapted varieties. Besides this, these markers can be used for marker-assisted backcrossing, pyramiding of the resistant genes, and they may be the starting point for gene isolation, followed by allele mining, and today also by allele editing. And this marker development in former time has been a quite time-consuming and laborious process, and the elder ones of us may remember techniques like RFLPs, genomic SSRs, and so on. Today, we have high-throughput chip technologies available, genotyping by sequencing, exome capture. We can use information from closely related species, and for the main cereals, wheat and barley, we have reference sequences today available, and techniques like CASP to use to develop then trait-specific markers which can be applied for low cost in cereal breeding. And in the next few minutes, I will give you some examples how we exploit this information for harnessing genetic diversity within my institute. For example, if we look at leaf, rust, and barley, 25 major resistant genes are known, but out of these, only RPH 15, 16, and with a limit to some extent RPH 7 are still effective in Europe. So some time ago, we screened the barley collection of the Serbian gene bank for resistance to Puccinia hordii, and in the end, we identified one landrace called MBR 1012, which turned out to be resistant to the most virulent strain in Europe, which is I80. And so we crossed this resistant line to the susceptible line scarlet, produced 100 DH lines, and were able to map this resistance genes on the telomeric region of the short arm of chromosome 1H, but you see still quite a large distance between the gene and the marker. So we used this population, Dragan Perovic in my group, to construct, based on the 9Ki select chip, a high-density map covering around 2,900 markers. And if we now have a look at this region, very quickly, quite closely linked genes for this new and effective resistant genes were developed. So in the next step, it was interesting to get information which genes are, which is the function of this resistance gene. So we constructed a high-resolution mapping population, tracing back to around 5,000 F2 plants. For marker saturation, we conducted GBS and used the bulks, which were analyzed with the 50K barley chip. And as you can see here, we were able to narrow down the interval to around 0.3 centimorgan. And if we now assign these markers to the reference genome of barley, it turned out that these covers around 0.4 megabases, and within these 0.4 megabases, all in all, 29 genes are located, and some of them are known to be involved in resistance and are now analyzed in more detail. And using these platforms is not only suited to develop markers for qualitative genes, but also to get more detailed information on quantitative resistances. In collaboration with Ernesto Igasturra's group from Spain, we identified within the Spanish barley core collection 2 QTL on chromosome 7HS and 7HL in the first mapping, comprising around 12 and 50 centimorgan. Using all the genomic information available, we were able to narrow down this year to 1.3 centimorgan, and by constructing a high-resolution mapping population, we were able to identify a cluster of nucleotide binding site leucine-rich repeats in this region of this quantitative trait locus, which is now under more detailed analysis. So these high-throughput marker technologies also facilitate association mapping, which in the end contributes to a faster harnessing of genetic diversity, because phenotyping and genotyping can be conducted simultaneously, and there is no need to construct biparental mapping populations. And this is done in barley for resistance to Pyrenophora teres. So out of 10,000 barley accessions, we selected 450. They were phenotyped, genotyped, and then association mapping was conducted in collaboration with partners from St. Petersburg. They were genotyped in Russia, in Belarus, in Quedlinburg, and also with specific isolates. And in a short period of time, you can see here, you can detect QTL, which are effective in all environments, like here on chromosome 6H, but also environmental or isolate-specific QTLs. Now, a technique to combine the advantages of biparental mapping and of association mapping is nested association mapping. And Klaus Pillen at the University of Halle constructed a nested association mapping population of crossed 25 barley accession, crossed to elite cultivar and back crossed 1 times. And we analyzed these 1,400 lines in 2 years field trials, and it turned out that with respect to the area under the disease progress curve and the reaction time, sufficient heritabilities were obtained to map QTL for these resistances. And as you can see here, just a brief overview, resistance to Pyrannophora teres is encoded by many small effect QTLs, and you see some QTLs which are effective as well with respect to the area under the disease progress curve, means the area, leaf area diseased, but also with respect to the reaction type. And if we now look into literature, we can see that we identified many QTLs in regions where already QTL effective against Pyrannophora teres are known. But there are also some new QTL, for example, here on chromosome 1H, chromosome 4H, and chromosome 7H. And these are now suited to broaden the genetic base of Pyrannophora teres resistance in barley. And this is easier because the exotic donor lines have been backcrossed already once to a highly adapted barley cultivar. Yeah, now let's come from barley to wheat. Leaf rust in wheat on the worldwide level is a very important disease. Let's say in Europe, it wasn't an issue till around 2006 because most cultivars carried the resistant gene LR37. But what you can see here, with more or less in one growing period, this resistance was completely overcome. So in my institute, we started to look in wheat relatives for resistance to Puccinia hordii, and within Triticum monococcum, we identified a race unspecific resistance which is pre-Haustorial. This means in the resistant genotype, the fungus is not able to create these Haustorial mother cells, which are essential for the fungus to fulfill its life cycle. And as you can see here, by inoculation with a mixture of different isolates, this resistance still remains to be effective. And now, to be sure, before we invest much in the analysis of the resistance, that we are not dealing with a resistance gene from the A genome already known, we isolated lines carrying all these genes with virulent isolates, and as you can see here, they all were susceptible, while the Triticum monococcum accession remained resistant. So to get more information on, or more detailed information on this resistance, we conducted microscopic and biochemical analysis, and what turned out here, you see in the resistant genotype already from the beginning, there is a higher peroxidase activity and also a higher chitinase activity, which can be also seen here in this microscopical photos. So, and to analyze this on the molecular level, we conducted MACE, massive analysis of cDNA ends, which is a technique where you can get information on differences in expression and on SNPs simultaneously. And to make a long story short, we collected samples at different time points, at very early stages of infection, and you can again see here that in the resistant genotype, some chitinases turned out to be upregulated at very early time points. The same holds true for many peroxidases, and besides these, different genes differently expressed between resistant and susceptible genotypes after infection were glucanases, pathogenous-related proteins, and the phosphoribonuclease. So now, in order to get information on the genetics of this pre-Haustorial resistance, we analyzed 184 F2 plants in F2 and then 10 progenies in F3 by artificial inoculation and counting Haustorial mother cells, and simultaneously we constructed a dart map comprising around 4,000 markers and 1,500 centimorgan. And you can see here, all in all, we identified 4 QTL involved in this prehistoric resistance. And if we now have a look which genes are located in the regions of these QTLs, we find again chitinases, pathogenesis-related proteins, and receptor-like kinases. And what is now done is to transfer these QTL marker-based from Triticum monococcum into Triticum aestivum in order to generate long-lasting resistance to Puccinia hordii. So, as we all know, the best marker is the gene itself. And we heard about map-based cloning from Christian already. And let's say in former times, map-based cloning has been really a laborious and time-consuming process because marker saturation, you had to use anonymous markers. Today, let's say you can use markers of which you know they are located in your region of interest. You can assign them to the reference map and you will easily identify candidate genes. And so I will now elucidate this with respect to the barley yellow mosaic virus resistance. This was one of the first genes cloned, resistant genes cloned in barley in collaboration with Andreas Graner. And so barley yellow mosaic virus is one of the most important diseases of barley in Europe because of yield losses, up to 40%, and these yield losses cannot be prevented by spraying chemicals, because these viruses here, from which different strains are known, are transmitted by the soil-borne Plasmodiophorid polymyxa graminis. So, and because the only way is growing resistant cultivars, tremendous efforts were undertaken to identify donors of resistance and to analyze genetics and to develop molecular markers. And as you can see here, many of recessive resistance genes and some dominants from Hordeum bulbosum were mapped within the barley genome, and 2 loci have been isolated using map-based cloning. And the isolation of the RYM number 4 gene took around 12 years, and it turned out that this locus encodes for a translation initiation factor. And most likely, this translation initiation factor, let's say, binds here to the VPG of the viral genome, and then translation starts. So this interacts at the very beginning of the virus, let's say, reproduction cycle. And next, RYM number 11 was isolated, and taking into account all the genomic information available at that time, this took around 4 years, and it turned out that resistance is due to a large deletion in a protein disulfate isomerases. These protein disulfate isomerases most likely act as chaperones, means they are involved in the correct folding of the viral protein. If they are not functional, the proteins are not correctly folded, ubiquinated, and degenerated. So, and if we now take into account that this gene interacts at the very first step of reproduction, and this in a later stage of the viral reproduction, then combining these resistant genes may lead to longer lasting resistances. So, and today, in addition, I showed you that many loci are known to code for virus resistance. So at the moment, isolation of these different resistant genes is in progress, and this is shown here for RYM number 13. You see again a high resolution mapping population, and marker saturation was constructed. Then these markers were assigned to the reference sequence of barley, and in the end it turned out that this region is equivalent to just 9 kb region, and in this 9 kb region there are only 3 genes, and one of these genes is known to some extent to be involved in virus resistance and will now be analyzed. So using this strategy, it's expected to isolate more of these resistance genes. So once the sequence of such a gene is known, it offers the possibility to conduct allele mining. And Ping Yang at the IPK in the group of Niyat Stein has done this. He has analyzed around 2,000 accessions for different alleles at the RYM number 11 locus. And all in all, he identified 4 different alleles, and all these alleles are non-functional. So let's say what you can now do, you can develop markers specific for these alleles and conduct breeding for resistance on the allele level. And if you know a sequence, what can of course also be conducted is today allele editing using the CRISPR-Cas system, maybe to create more powerful alleles at these different loci, because not all of these, for example, for RYM number 4, it's not effective against all strains of barley mild, barley yellow mosaic virus. So coming to an end, I hope I could show you that the availability of dense marker maps, high-throughput genotyping platforms, physical maps and genome sequences, as well as of high-throughput expression profiling techniques, facilitates an enhanced marker development and isolation of resistance genes, thereby leading to a deeper understanding of resistance and the transfer of marker-based selection to the allele level. Knowledge on resistance gene sequences will facilitate efficient allele mining and the targeted editing of respective alleles in the future by TALENs and CRISPR-Cas. And this, all in all, will lead to a more directed use of plant genetic resources in wheat and barley breeding, thereby broadening the genetic base of resistance. And in the end, I'd like to thank especially my group at the JKI, who produced all of the results I have presented here, my former group under the supervision of Wolfgang Fried at the University of Gießen, where the foundations for the isolation of these resistance genes, RYM number 4, RYM number 11, were laid. Andreas Graner and Niyat Stein from the IPK for the fruitful collaboration with respect to isolation of resistance genes. Klaus Pillen from the University of Halle for the collaboration on the nested association mapping. Ernesto Igatua and his group from Spain for the mildew data, and the Pyronophora terrace work is done in close collaboration with Brian Stephenson and Olga Afanasenko. And at least I'd like to thank these organizations for financial support and you for your attention.
We have a pretty good amount of time. Questions? Young people? I have one question in the comments. You can see this research is it's a long processing from the low map. 10 years ago, you never stop. You just add more markers and add more markers and then short that QTL region. Finally you found this gene. That's good work. And I like just those young people here to know it's not something you can do it like that fast. It is a continual work, and hopefully young people can
Let's say I completely agree with you. Let's say the problem today is not the marker saturation and all of this. I think if you do marker map-based cloning, you can do it. You can isolate a gene within 3 years. The thing is that you really have to combine all the steps. Means screening large gene bank collections to identify donors of resistance. Do test for allelism, that you do not work on a gene which has, for example, already been isolated. No? And then you can start all this procedure. But I think if you really make use of, let's say, the genomic information possible, and also if you have a trait which is quite easy to phenotype, no? Then you can also use direct sequencing, what Jonathan Jones explained yesterday, to get quite rapidly to a candidate gene, and also to do RANSAC. But let's say with respect to this virus resistant gene, they are not classical resistant genes, as you learned. So I think their map-based cloning is still the right strategy to isolate these resistant genes. But employing all the genomic data we have available, it has really become much more easier and much more directed than in former times.
You can see that Frank also mentioned the phenotype. Remember, phenotype is even more important part of work.
Yes, let's say I'm quite lucky. Let's say I work at an institute which was the former Institute of Phytopathology of the GDR. And so I have a very well trained phytopathologists around me. So phenotyping is really crucial, because if you make any mistakes in phenotyping, you will never isolate the gene. You will not land or not identify candidate genes. So that's crucial. And let's say what we also do, what Andreas has shown today, let's say we are investing to some extent in automatic phenotyping and in hyperspectral imaging to get very reliable results.